Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 40. The topic is adenosine triphosphate. Discussing agenda for the topic will be ATP powers, cellular work, the structure and hydrolysis of ATP, and how ATP provides energy that performs work, and finally the regeneration of ATP. Lecture outcome. After watching and listening this lecture, students will know about the structure of ATP, the work it performs, and how it is regenerated. Introduction. ATP powers cellular work by coupling exergonic reactions to endergonic reactions. ATP is one of the most important molecules in biochemistry. It's a remarkable molecule. It's an energy carrier, but at the same time, it carries energy for a very short time, and as soon as it is synthesized, it provides some energy to perform some work in the form of contraction and relaxation of the muscles, or the movement of molecules across the plasma membrane, or the synthesis of molecules in anabolic reactions. So a cell does three main kinds of works where ATP is consumed within the cell. The first is chemical work, and it is the pushing of endergonic reactions, the reactions which requires the input of energy, because endergonic reaction does not occur on their own. They are non-spontaneous. They doesn't occur on their own and requires the input of energy. And these reactions are quite often involved in the anabolic pathways, where the synthesis of molecules, the synthesis of polymers from monomers takes place. The second type of work is transport work, which is concerned with the movement of molecules across the plasma membrane against the direction of spontaneous movement, which refers to the fact that the movement is against the concentration gradient. The third is mechanical work, which refers to the beating of cilia, the contraction of muscle cells, and the movement of chromosomes during cellular reproduction. Remember the process of mitosis or meiosis, when in metaphase stage the chromosomes are arranged on the equator, and then the spindle fibers from the poles emerges, and the spindle fibers attaches to the kinetochore region, which is present on the centromere, and as the depolymerization of the spindle fibers takes place, the chromosomes are pulled towards their respective ends. So this process also requires ATP. Now, a key feature in the way cells manage their energy resources to do this work is energy coupling. Energy coupling means wherever energy is used, means wherever an endergonic reaction is involved, alongside that an exergonic reaction will be occurring. The energy liberated by the exergonic reaction will be incorporated into the endergonic reaction. So these endergonic reactions and exergonic reactions, they occurs simultaneously. They are just like the two rail tracks, so they occur side by side. Now ATP is responsible for mediating most energy coupling in cells. So wherever a process is in need of some kind of input of energy, then this process is referred to as inorganic process, and alongside that process the ATP molecule will be broken down and the energy liberated in this exergonic reaction will be incorporated in that reaction. Now, how this remarkable molecule looks like. So ATP, which is abbreviated as adenosine triphosphate, it contains a sugar, a ribose sugar, and a nitrogenous base, which is adenine, and 3 phosphate groups that are arranged in a sequence, and these are high energy phosphate groups. Their bonds store high energy, especially this third phosphate group. In addition to its role in energy coupling, now ATP is also a source of energy, and it is also one of the nucleoside triphosphates used in the synthesis of RNA. The bonds between the phosphate group of ATP can be broken down by hydrolysis, that is in the presence of water. These bonds can be broken and energy is liberated. The ATP, when loses one of its phosphate, becomes ADP, and the energy liberated is used to perform work. When the terminal phosphate bond is broken by addition of a water molecule, a molecule of inorganic phosphate, abbreviated as PI throughout this book, throughout this Campbell's Biology 12th edition book, the inorganic phosphate will be represented as PI. So this inorganic phosphate leaves the ATP molecule. The remaining molecule is ADP with 2 phosphates, and in this process energy is liberated. How much energy is liberated? 7.3 kilocalories per mole of ATP hydrolyzed. Remember, this 7.3 kilocalorie is not for a single ATP, but it is for 1 mole of ATP molecules hydrolyzed. Now this is the overall reaction, how the ATP is hydrolyzed in the presence of water molecule. The terminal phosphate bond is broken down and one of the phosphate is released as inorganic phosphate. The remaining molecule has 2 phosphates, and in this process energy is liberated. How much energy? 7.3 kilocalories per mole. So this is an exergonic process, and this process occurs alongside an inorganic reaction, and the energy which is liberated here is incorporated in that inorganic reaction. So this is the coupling. Now the delta G usually refers to the energy which is available to perform work. It is known as Gibbs free energy or simply free energy. It is the energy which is available for performing work, and when the value of delta G is positive, it means that this energy will be supplied to reactants to react with each other, and when the value of delta G is in minus, in negative, it means that this energy is liberated in a reaction. Now remember that this 7.3 kilocalories per mole is equal to 30.5 kilojoules per mole. The units are different, therefore the values are different. This is the free energy change measured under standard conditions. In the cell, conditions do not conform to standard conditions, primarily because reactants and product concentration differs from 1 mole. Now, for example, when ATP hydrolysis occurs under typical cellular conditions, the actual delta G is about minus 13 kilocalories per mole. That is 75% greater than the energy released by ATP hydrolysis under standard conditions. So the conditions inside a cell are not standard conditions, therefore the energy liberated from the hydrolysis of ATP is much lesser. You can say it is 75% lesser than the energy which is liberated under standard conditions. Now, because their hydrolysis release energy, the phosphate bonds of ATP are sometimes referred to as high energy phosphate bonds, but the term is misleading. Why? The phosphate bond of ATP are not unusually strong bonds, as high energy may imply. Rather, the reactants ATP and water themselves have high energy relative to the energy of the products ADP and PI. So here the reactants have high energy as compared to the products, and the products are at a lower energy level. Where this energy goes, which is liberated by the reactants, this energy is available to perform work. Now, the release of energy during the hydrolysis of ATP comes from the chemical change of the system to a state of lower free energy, not from the phosphate bonds themselves. So ATP is useful to the cell because the energy it releases on losing a phosphate group is somewhat greater than the energy most other molecules could deliver. That's why, because of that reason, it is regarded as the energy currency of the cell and a remarkable energy carrier. But why does this hydrolysis release so much energy? So now the question arises that ATP carries energy and it can deliver energy when it splits, but from where this energy comes, and why this process of hydrolysis releases so much energy? So the answer for that is there are 3 phosphate groups in ATP, and all these phosphate groups are negatively charged, and remember that the same charges repel each other, so there is a strong tendency of repulsion between the 3 phosphate groups. These like charges are crowded together, and their mutual repulsion contribute to the instability of this region, that is the region where phosphates are located. So the triphosphate tail of ATP is the chemical equivalent of a compressed spring. Just like a compressed spring has enough potential energy that if it is liberated, then it can expand and the potential energy can be released. When ATP is hydrolyzed in a test tube, the release of free energy merely heats the surrounding water. In an organism, this same generation of heat can sometimes be beneficial. So, the hydrolysis of a single ATP releases a small amount of energy if you observe it, and by hydrolyzing an ATP in a test tube, but even that small amount of energy when liberated inside a living system is beneficial. For instance, the process of shivering uses ATP hydrolysis during muscle contraction to warm the body. So shivering is quite often associated with the cold weather where the process occurs, and in this process the energy which is released by the contraction of the muscles warm the body. In most cases in the cell, however, the generation of ATP alone would be inefficient and potentially dangerous use of a valuable energy resource. Heat, you already know from the second law of thermodynamics that the energy which is liberated in the form of heat is usually a loss of energy, because the heat released in the environment increases the entropy of the system, the randomness of the system, the disorder of the system, and the heat cannot perform work. And you can put that sentence in a bracket too, that heat can only perform work when there is a temperature gradient. So the liberation of energy in the form of heat is actually a waste of energy. Instead, the cell's protein harness the energy released during ATP hydrolysis in several ways to perform the three types of cellular work. So the liberation of energy by ATP, if takes place in a test tube, then it will warm the test tube. Similarly, during shivering, the ATP molecule is hydrolyzed and it warms the body. So if the generation of energy by the ATP is only in the form of heat, then this is a waste of energy, because the heat cannot be utilized to perform work. However, there are certain mechanisms in the body which utilizes the energy released by ATP, and this energy is utilized to the benefit of the body and to perform different types of work like chemical, transport, and mechanical. So this energy which is released is not wasted as heat and incorporated in the performance of these types of works. For example, with the help of a specific enzyme, the cell is able to use the high free energy of ATP to drive chemical reactions that is by themselves are endergonic. So there are specific enzymes which are present, and with the help of those enzymes the cell is able to use the energy released after the hydrolysis of ATP for performing work. Now, if the delta G of an endergonic reaction is less than the amount of energy released by ATP hydrolysis, then the two reactions can be coupled so that overall the coupled reactions are exergonic. Now suppose the ATP is releasing 7.3 kilocalories per mole energy, and there is another reaction going on which requires the input of energy, and the input of energy is less than the one produced by ATP. Suppose it is 3.3 kilocalories or 4 kilocalories or 5 or 6, but less than the energy produced by the hydrolysis of ATP, then both these reactions are coupled and the energy released by the ATP increases the energy of the reactants. It brings the reactants to a transition state, and then they can convert into products easily. This usually involves phosphorylation. So how the proteins which are present in a cell utilizes the energy liberated by the breakage of ATP molecule for their benefit. So actually the process of phosphorylation is involved. Suppose there is an endergonic reaction going on, and this endergonic reaction needs the excitation of the reactant molecules. Then what happens? The ATP molecule loses one of its phosphate, and this phosphate phosphorylates one of the intermediates, one of the reactants. As a result, the reactants become in transition form, and the reaction can be proceeded forward. In the next slide we will see the example of that. The recipient molecule with the phosphate group covalently bonded to it is then called a phosphorylated intermediate. The key to coupling exergonic and endergonic reaction is the formation of this phosphorylated intermediate, which is more reactive, that is less stable with more free energy than the original unphosphorylated molecule. So now we will study this example in graphical representation form. Now the reaction of glutamic acid with ammonia, it is a reaction which requires the input of energy, and the ATP molecule must provide this energy. So how the ATP molecule provides energy? ATP loses one of its phosphates. The phosphate becomes part of the glutamic acid. Now this glutamic acid is in transition, in unstable stage, and now the reaction is more feasible. Now ammonia will combine with this activated or unstable phosphorylated intermediate, and the products will be formed. So the first conversion of glutamic acid to glutamine, how it takes place? It must react with ammonia. This process is an endergonic one, requires the input of energy. Remember, if the value of delta G is positive, then it means the process is non-spontaneous and requires the input of energy. So first the ATP molecule loses one of its phosphates. This inorganic phosphate attaches to the intermediate, the glutamic acid, and then the glutamic acid is in excited state, it is in less stable state, and now the reaction can proceed forward. It can readily react with ammonia and form glutamine, and the ADP and phosphate, inorganic phosphate, is recovered back. So you can see here now the input of energy required for this process is 3.4. So it is an endergonic reaction, and because ATP was hydrolyzed in this process, so the ATP released that much energy. The hydrolysis of one ATP molecule releases that much energy. So what will be the net delta G? The input of energy was 3.4. The energy supplied by ATP was 7.3. So 3.9 kilocalories of energy was liberated by this process and is available to perform work. Now just go back to that first sentence. If the delta G of an endergonic reaction is less, so in that case the delta G was 3.4 something, and the energy liberated by ATP was 7.3. So then the two reactions are coupled, as you saw here. The energy required was 3.4, the energy supplied to it was 7.3. So the net delta G which is still available to perform work is 3.9. Now how ATP drives energy that performs work. So we continue with our previous heading. Transport and mechanical work in the cell are also nearly always powered by the hydrolysis of ATP. In these cases, ATP hydrolysis leads to a change in a protein's shape and often its ability to bind another molecule. So in the previous case, the ATP drives a chemical reaction by binding to one of the reactants and converting the stable reactant into unstable form. So how ATP drives transport and mechanical work? In this case, the hydrolysis of ATP changes the shape of a protein and its ability to bind another molecule. Sometimes this occurs via a phosphorylated intermediate as seen from the transport protein in figure 3.A. So here you can see now how the transport work takes place, means how the molecules from the extracellular matrix are transported inside into the cell. Remember, if ATP is used, then this process will be against concentration gradient. And now how ATP is catalyzing this process. ATP helps in the change of one of the proteins, and this change enables the protein to carry out the transport work. So this protein is in this form, and then so these are transport proteins. They change their conformation, and these conformational changes enables them to perform their activity. So this is the example of this. ATP hydrolysis leads to a change in protein's shape and often its ability to bind another molecule. Now, in most instances of mechanical work involving motor proteins walking along cytoskeletal elements, a cycle occurs in which ATP is first bound non-covalently to the motor protein. So recall from the cytoskeleton topic where there are motor proteins that are responsible for walking over the cytoskeletal elements and transporting materials from one organelle to another organelle within the cell, and these proteins are called the motor proteins. So this is a motor protein, this is the cytoskeletal element, and this motor protein is holding a vesicle which is destined towards some other organelle or towards the cell membrane, and this motor protein a kind of walk along the track laid by the cytoskeletal elements, and this process is catalyzed by the ATP molecule. So this is the intracellular transport mechanical work. Transport and mechanical work in the cell are also nearly always powered by the hydrolysis of ATPs. In these cases, ATP hydrolysis leads to a change in protein's shape and often its ability to bind to another molecule. In most instances of mechanical work involving motor proteins walking along cytoskeletal elements, a cycle occurs in which ATP is first bound non-covalently. So I think that's a repetition from the previous slide. Next, ATP is hydrolyzed and releasing ADP and inorganic phosphate. So another ATP molecule can then bind and it can power the motor protein and help in transportation. At each stage, the motor protein changes its shape and ability to bind to the cytoskeleton, resulting in movement of the protein along the cytoskeletal track. The phosphorylation and dephosphorylation promote crucial protein shape changes during many other important cellular processes as well. So I hope you have got a bit of concept from the last 3 slides how ATP catalyzes cellular work. ATP usually do so by phosphorylating a protein which then changes its conformation, or it phosphorylates one of the intermediates of a chemical reaction and makes it unstable and readily reactable. Now another important thing is that ATP is a remarkable molecule. It stores energy and it can perform work, but the problem with ATP is that it stores energy for a very short period of time. As soon as it is formed, it is hydrolyzed again. So this cycle occurs over and over again continuously. ATP is formed, then it loses its phosphate, becomes ADP, then it picks phosphate, becomes ATP again. So this process occurs continuously in a cycle. Now, as organism at work uses ATP continuously, but ATP is a renewable source that can be regenerated by the addition of phosphate to ADP. The free energy required to phosphorylate ADP comes from exergonic breakdown reactions. Catabolism in the cell. Catabolism is the breakdown of glucose molecule or other molecules which releases energy, and this energy is incorporated in the third phosphate bond in ATP. This shuttling of inorganic phosphate and energy is called the ATP cycle, and it couples the cell's energy yielding exergonic process to the energy consuming endergonic ones. So what happens that at one point in a cell a catabolic reaction is going on which releases energy. This energy is incorporated into ATP molecule. Then ATP molecule couples another process which requires the input of energy. So at that instant the ATP molecule itself is hydrolyzed. So in this way the catabolic and anabolic processes or the exergonic and endergonic reactions occurs in the cell. I repeat that sentence again. Wherever in a cell a catabolic process is going on, then this catabolic process will release energy, and this released energy will be used to form ATP. And then wherever in other areas of the cell, if an endergonic process is going on, then this ATP which was synthesized previously shifts to that particular area, and it is broken down and the energy liberated is incorporated into endergonic reactions. The ATP cycle proceeds at an astonishing pace. So this cycle occurs very, very fast. For example, a working muscle recycles its entire pool of ATP in less than a minute. So in less than a minute, all the ATP molecules which are there in the muscle loses their phosphate group, delivers energy, and then again they are recycled into ATP form. So the process is that much faster. Therefore we say that ATP carries energy, but it carries energy for a very short period of time. The turnover represents 10 million molecules of ATP consumed and regenerated per second per cell. So just imagine this value, 10 million molecules of ATP will convert into ADP, will lose their phosphate, and then again will be regenerated to ATP in just 1 second per cell. If ATP could not be regenerated by the phosphorylation of ADP, humans would use up nearly their body weight in ATP each day. So the consumption of ATP is of great importance because it runs the bodies, and therefore this regeneration of ATP is important, otherwise the effects will be catastrophic on the body. This is the ATP cycle. ATP is synthesized and then hydrolyzed back into this form, then again synthesized. So this process is endergonic, and from where this energy comes? This energy comes from other catabolic reactions which are exergonic, and then when ATP is broken down, again energy is released, and this energy is incorporated in some endergonic reactions. So because both directions of a reversible process cannot be downhill, the regeneration of ATP from ADP and inorganic phosphate is necessarily endergonic, because ATP holds energy, so this energy is provided when ADP combines with inorganic phosphate. So this formation of ATP is endergonic and hydrolysis of ATP is exergonic. Here is the overall reaction. ATP molecule combines with inorganic phosphate and forms ATP, and then this ATP molecule in the presence of water is hydrolyzed back and converted into this form. How much energy will be liberated when ATP is broken down? 7.3 kilocalories per mole under standard conditions. But remember that inside the cell, the cellular conditions are not standard conditions, so the energy produced is much less. Recall from the previous slides that 63% value. Since ATP formation from ADP and inorganic phosphate is not spontaneous, this is an endergonic reaction and it will not occur on its own. It requires the input of energy. The catabolic pathways, especially cellular respiration, provides the energy for the endergonic processes of making ATP. So wherever there is an exergonic reaction going on, there is a catabolic process going on, then the energy liberated in this catabolic process is used to phosphorylate ADP and form ATP. So plants also use light energy to produce ATP. Thus, the ATP cycle is a key player in bioenergetics, functioning as a revolving door. I hope you know about the revolving door through which energy passes during its transfer from catabolic to anabolic pathways. So the catabolic and anabolic pathways cannot occur side by side. Actually, this ATP is the intermediate. It picks the energy from catabolic pathways and deliver this energy to the anabolic ones. This was the end of the lecture. Thank you and Allah Hafiz.